So here we are in chapter 2, which is a review of chemistry, which is something you guys are all probably excited about. Since physiological processes are based on chemical reaction, it's important to understand a general understanding of chemical principles. So we'll look at atoms, ions, chemical bonds, and we'll finish up with DNA. And again, this lecture, normally I would probably spend 2 to 3 hours on in class, but since no one is going to want to watch a video that long, this will be a shortened version of that. So again, I would encourage you guys to read your textbook, work through the end of chapter questions, and if you still have questions, to please let me know. Hopefully some of this will be a review because most of you should have taken chemistry or are in chemistry right now. Let's start with an atom. It's the smallest unit of an element. The nucleus has protons, which are positively charged, and uncharged or neutral neutrons. Orbiting electrons have the negative charges. An atomic mass is equal to the number of protons plus the number of neutrons, and an atomic number is equal to the number of protons. You should know everything on this slide, the basic understanding of an atom, what's in the nucleus, what surrounds it, what are the charges of each of the particles, and what does atomic mass mean, and what does atomic number mean. This is a look at the protons and neutrons in the nucleus with the electrons circling it. Electron orbitals are shells of energy that surround the nucleus, and electrons fill these shells. The first shell holds 2 electrons, and each shell thereafter holds 8 electrons. Atoms are most stable when their outer shell is filled. So electrons in unfilled outer shells will participate in bonding, and these are called valence electrons. So these are atoms commonly present in organic molecules in the body. Their atom, the symbol, the atomic number, the atomic mass, the electrons, and then the number of chemical bonds. Isotopes are atoms of the same element but with different numbers of neutrons. So their atomic number is the same, but the atomic mass is different because, again, atomic number is the number of protons, but their atomic mass will be different because there's a different number of neutrons. Some isotopes are radioactive, so they're helpful in medical testing and research. Chemical bonds are formed when electrons and atoms interact. So the number of bonds an atom can form is determined by the number of valence electrons. So since hydrogen just has 1 electron, it needs 1 more to fill an inner shell so it can form 1 bond. Carbon has 6 electrons, 2 in the inner shell and 4 in that second outer shell, and it needs 4 more to complete its shell. So it will form 4 bonds. Oops, sorry about this, 1 second. So this is just a look at covalent bonds. Valence electrons are shared because energy levels overlap. This is what covalent bonds will look like with their electrons overlapping. More examples of covalent bonds between atoms overlapping with their electrons. Water is a polar molecule. That means one side is more negatively charged than the other. It's a good solvent, which means substances will dissolve in it. It can form strong hydrogen bonds, and polar molecules may dissolve in water. And we call anything that dissolves in water hydrophilic or water-loving, but non-polar molecules will not dissolve in water, and we call them hydrophobic or water-fearing. So this is what water looks like. H2O, the O is in the center with the hydrogen on the other side, and you can see how oxygen has a more negative charge. So the negative charge will be on this side of water, and the positively charged hydrogens are over here. So it kind of has this polarity to it where one side is more negative and one side is more positive. Ionic bonds, one atom gives electrons to another so that both have filled valence shells. Cations and anions will attract each other. Cations are positively charged, which is the electron donor, and anions are negatively charged because they are the electron receiver. So this is a look at an ionic bond. A great example of this is sodium chloride, which is table salt. Most ionic compounds dissociate when they are dissolved in water. And this is a look at how a water molecule will separate the sodium and chloride from each other and surround it. So it'll dissociate the salt into a sodium and a chloride ion. Hydrogen bonds are weaker bonds formed between two polar molecules. They form between amino acids on a protein. And this hydrogen bonds, if you remember anything about hydrogen bonds, is that they will hold the two strands of a DNA molecule together. They're between the base pairs. So these are look at hydrogen bonds between water molecules. Acids, bases, and pH, some water molecules ionize to form free hydrogen ions or protons, which we call H+, and hydroxide ions, which we call OH-. When this happens, if there's the same number of ions, the solution is neutral, and a neutral solution is said to have a pH of 7. When a solution has more hydrogen ions than hydroxide ions, this is called an acid, and its pH is below 7. We call it a proton donor because it'll have more of these hydrogen ions or protons to donate. When a solution has more hydroxide ions, we call it a base, and its pH is above 7, also called alkaline, and we call this a proton acceptor. Here's the pH scale. It runs from 0 to 14, where 0 is the strongest acid, 14 is the strongest base, and a pH of 7 is neutral. Here's a look at acids and bases and their relative concentrations of hydrogen ions and hydroxide ions. Buffers stabilize pH in a solution, and I'll let you read through how buffers stabilize the pH. If blood falls below 7.35, the condition is called acidosis, and if it rises above 7.45, the condition is called alkalosis, so more basic. Most organic molecules will always contain carbon and hydrogen, and I'll let you guys read through the rest of this slide. Carbon bonding is seen here between ethane and ethylene. This is a look at how we draw bonds. If there's a single dashed line, that's a single bond, and if there's a double dashed line, that means a double bond. So again, most organic molecules will contain carbon and hydrogen. Hydrocarbon structure, carbons are not shown but are understood to be at the corners. Some will show double bonds. Carbon chains and rings form backbones for more reactive groups of atoms called functional groups. And functional groups allow organic compounds to be classified into groups, which provide unique chemical properties. So if we look at hexane, all in a line separated out here, hexane, you know, a hexagon has 6 sides, so a hexane molecule has 6 carbons. Cyclohexane is the hexane molecule, but in cyclic form or a circle, and oftentimes we'll get rid of the carbon and we'll just label it with a corner, and it'll be assumed that a carbon is at the corner. And a benzene ring is a 6-sided cyclic structure with 6 carbons and 6 hydrogens, but there's double bonds shown. Here are common functional groups, which you would learn about in organic chemistry if you ever get that far. I loved organic chemistry, but that makes me a nerd, so we'll move on from this. A ketone has a double bond attached to oxygen. This is a look at an organic acid. It always has this hydroxide group. An alcohol always has an OH group on the end, and an aldehyde is double bonded to oxygen and a hydrogen, but we'll move on from here. Stereoisomers have exactly the same atoms arranged in the same sequence, but they differ in their spatial organization of their functional groups. And you can see that here, cis and trans 2-butene, their functional groups, the cis, they will be on the same side, and the trans, they'll be on opposite sides. Carbohydrates and lipids, these are organic molecules that contain carbon, hydrogen, oxygen in that 1 to 2 to 1 ratio. And these will serve as a major source of energy. They include sugars and starches. They will often end in "-ose" at the end, and this will denote a sugar. So examples are monosaccharides, which is a simple sugar, 1 carbon ring, like glucose, fructose, galactose. Disaccharides are 2 monosaccharides joined together by a covalent bond. So we get sucrose, maltose, and lactose. And these are just examples of monosaccharides. Feel free to look more into this on your own. Polysaccharides are several monosaccharides joined together. Starch is the plant form of sugar, glycogen is the sugar storage in an animal cell, and cellulose makes up cell walls. So here's glycogen, it's the storage form of glucose. Dehydration, synthesis, and hydrolysis. Hydrolysis breaks bonds between monosaccharides. It adds water and split the molecule. And this is a look at dehydration synthesis. Hydrolysis adds water. Lipids consist of nonpolar hydrocarbon chains and rings that makes them hydrophobic or insoluble in water. We have triglycerides, which are fats and oils. Fatty acids are long nonpolar hydrocarbon chains with carbonyl at one end, which is a carbon, oxygen, and hydroxide group. And we're going to talk about saturated and unsaturated fatty acids here, because you'll see this commonly in food labels. So if we look at an unsaturated fatty acid, you can see this double bond. And the unsaturated fatty acid, the double bond causes, this doesn't show it here, but the double bond causes this chain of fatty acid to kind of be kinked or bent. And the saturated fatty acid doesn't have any double bonds. Because a saturated fatty acid doesn't have any double bonds, this molecule is completely linear. And the saturated fatty acids can stack on top of each other and be harder to break down. So they say saturated fat is worse for you because it's harder for your body to break it down because they're all linear molecules stacked on top of each other. Whereas an unsaturated fat, these double bonds cause the molecule to kind of kink or bend, so they're unable to be stacked on top of each other. And this kind of shows that double bond where as it's cis or trans, just making it harder for them to be stacked on top of each other. Triglycerides then are shown here. This kind of shows saturated and unsaturated fats, the ketone bodies. I'm going to move over this for right now. More on phospholipids. I'll let you guys read through here. Phospholipids are lipids with a phosphate group, which makes them polar, and they're a major component of cell membranes as a double layer with their hydrophilic phosphates pointing outward on each side and hydrophobic fatty acids and glycerol pointing inward. That might be a quiz or a test question coming up about phospholipids. So you can see their hydrophilic head and hydrophobic tail. Micelles in water is just showing how water will surround and be attracted to the hydrophilic or water-loving head of the fatty acid. And here are steroids, and it's structurally very different from a triglyceride, but it's nonpolar, so it's considered a lipid. And my daughter just walked in, so 1 second. Can you ask Poppy to help me? Go ahead. Cholesterol is a steroid used as a precursor to steroid hormones, like testosterone and estrogen, which we'll talk about. So here are what steroids look like. Prostaglandins, it's a type of fatty acid with a cyclic hydrocarbon group. So here are prostaglandins, and they will help regulate blood vessel diameter during ovulation, uterine contractions, blood clotting, very important in the body as well. Proteins then are composed of an amino acid chain. There are 20 different amino acids that can be combined in an endless number of ways. Amino acids are charged, so they'll attract each other, and the sequence of amino acids in a chain is determined by your DNA. So your DNA is a code for coding for different proteins and amino acids. Here are an example of some of the amino acids. Nonpolar and polar, and they just have different types of functional groups attached to them. When amino acids are joined, a hydrogen is stripped. I'll let you guys read through this to make a peptide bond, and they'll eventually make the primary structure of a protein when their amino acids are added together. Making a protein, the protein structure, it's a chain of amino acids. It's called a polypeptide chain. It can vary in length. You can see there from 3 to 4,500 amino acids. And this chain is called the primary structure. Weak hydrogen bonds may form, and and this could form an alpha helix or a beta fold, and this is called the secondary structure of a protein. And you can see here how the secondary structure will form this helix. And then we get to tertiary and quaternary structure as well. And a good example of quaternary structure, this shows hemoglobin, which should be a little familiar from anatomy, and we'll go over more why hemoglobin is important. Here's protein structure into 3D shape, tertiary structure, and then into quaternary structure. And examples of this are hemoglobin, which are 4 polypeptides, and the hormone insulin. Conjugated proteins are where sometimes proteins will be combined with other molecules to become functional, where we'll get a glycoprotein or a lipoprotein. You can see examples where each would be located in the body. Here, more conjugated proteins. Protein functions. It's important to know the functions of proteins. They help in structural ways. They are enzymes, they're antibodies, they're receptors, and they're also carriers. More protein functions, they make up collagen and elastic fibers. This is a look at loose areolar connective tissue. Nucleic acids then, these are the building blocks for nucleic acids or nucleotides. So if we start with nucleotides, they're composed of a 5-carbon sugar, a phosphate group, and a nitrogenous base. And it'll be guanine, adenine, cytosine, thymine, or uracil. And it'll be deoxyribose or ribose, depending on if it's DNA or RNA. So the sugar in this molecule of DNA is called deoxyribose, and it can form with these nitrogenous bases, guanine, thymine, cytosine, and adenine. And then these are where the complementary base pairs will join up with each other. The 2 chains of DNA will be twisted, forming a double helix. And DNA, like I mentioned before, is the basis for the genetic code. So this is a look at deoxyribonucleic acid and the 4 bases. And the pairing guanine always pairs with cytosine, and thymine will always pair with adenine. And again, the bases will be formed between the 2 with hydrogen bonds, which means they're weak, so they'll be able to be dissociated so DNA can replicate itself, and they'll wind around each other to form a double helix. RNA is similar to DNA except has a ribose sugar. It's single-stranded, and it will have uracil instead of thymine. So this is the difference between DNA and RNA, and you should know these differences. Ribonucleic acid, there's 3 types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Other RNA-related molecules are ATP, CAMP, and NAD/FAD, which are also extremely important and we'll get through and cover. And that's the end of chapter 2. Thanks for listening, guys. I'll see you for chapter 3.